Hi everyone, welcome to Neuroscience for Non-Majors. I'm looking forward to meeting all of you tomorrow evening. I hope everything's going as well as it can for all of you during these incredibly stressful times. Well, tomorrow we'll go over the syllabus more in depth and figure out office hours and stuff like that during class. I want to start this first lecture with a brief introduction of myself and a short overview of the course goals and structure. I promise we'll get to the neuroscience really soon though. My name is Arula Ratnakar, and I'm in my senior year of undergrad studying biology with a neuroscience concentration and an architecture minor. Long story short, I used to be an architecture student, then I got into set design, which led me to screenwriting, then science fiction writing, and finally science. I ended up liking science so much that I found myself self-studying neuroscience instead of doing my architecture schoolwork, so I decided to switch majors. I still remember the struggle during my time of self-studying though, and what difficulties and questions and insecurities about starting to study science I had back when I didn't even really know what exactly DNA was. That's why, after I switched majors and learned enough biology and neuroscience to feel comfortable teaching the material, I decided to make this course for anyone who wants to learn this stuff, but doesn't know where to start. Because I remember how intimidating it was, and I want to make it the opposite of intimidating for you. While this is a very fast-paced class, my goal is to provide you with as much information as I can in the most accessible manner possible, and for you to ask questions, work through example problems, and discuss whatever interests you further and whatever you don't understand during the in-class meetings. Whether those questions are what exactly is a protein, or can we use an autoencoder to simulate this brain system? There are no exams in the course, and the homework is designed to be low stress and fun, and is graded on completion, not correctness. This class is for everyone, and there's no such thing as a stupid question. I encourage you to reframe what you think of as stupid questions and start thinking of them as naive questions instead. One professor I had said something like this once: while most naive questions will simply result in your understanding something better, sometimes naive questions are what win Nobel Prizes. Either way, you end up better off for asking them. By the end of this course, I hope that you will have a solid understanding of fundamental neuroscience concepts, be able to think creatively about future directions for neuroscience research, and understand the applications of neuroscience in a wide variety of fields, from actual neuroscience to science fiction writing to philosophy to artificial intelligence. Outside of class, as homework, you will be required to watch the pre-recorded lecture video each week before class and complete one short homework assignment each week. In class, we will go over the material together, work through specific problems, and answer questions you might have, as well as discuss what interests you about the week's topics. There will also be two projects, one at mid-semester and one during finals, but I will go over that more tomorrow. It's time to get into the neuroscience. All right, I'm going to share my screen. I have a presentation for you. Okay. I hope it loads. I was having issues loading it, but okay. So, in this lecture, we will learn about the structure of a neuron, ion concentration, electrical force and diffusion, equilibrium potential, ionotropic receptors, action potentials, and presynaptic and postsynaptic roles in a synapse. I'll start with a basic outline of how your brain works at the cellular level, and then we will revisit this outline and fill in the details. But basically, your brain contains around 86 billion cells called neurons that communicate with one another. And this communication is what allows you to think and feel and perceive. The way that neurons communicate is through these things called synapses, which are small gaps between the sending neuron and the receiving neuron. There are nearly 100 trillion synapses in the brain. At the synapse, the sending neuron releases chemicals called neurotransmitters to the receiving neuron. This causes the receiving neuron to create an electrical signal which allows it to release its own neurotransmitter to other neurons, thus permitting a chain reaction, communication. All right, let's fill in the details now and look at the structure of the neuron itself. This drawing does not look exactly like what the neurons in your brain look like, but it is useful for the purpose of illustrating the different parts of the neuron, which the neurons in your brain have. These things sticking off the left-hand side of the neuron in my illustration are called dendrites, and the dendrites contain receptors for neurotransmitters. The round portion is the cell body or soma, and it contains the nucleus of the cell. This projection here is called the axon, and it's what the electrical signal propagates through. The axon is wrapped with an insulating sheath made up of other cells called oligodendrocytes. And this sheath itself is called myelin, and it allows the electrical signal to travel along the axon quickly. The axon ends in these little bumps in my drawing called synaptic terminals, which contain bubbles of fat called vesicles, which are full of neurotransmitter. The synaptic terminal is what releases that neurotransmitter to other neurons. What separates the extracellular fluid outside the neuron from the cytoplasm that's in the interior of the neuron is a cell membrane. Within this membrane are all sorts of proteins that act as channels for specific ions. Ions are atoms that have lost or gained electrons, giving them a net electric charge. And the channel proteins allow the ions to flow into and or out of the cell, consequently changing the overall charge inside the cell. This ion flow through the channels, which can be open or closed, is crucial for allowing neurons to ultimately send their electrical signals when that neurotransmitter we were talking about earlier is released. Concentrations of particles, such as ions, generally want to move from high concentrations to low concentrations through any like channel or hole in a divider or whatever between the two concentrations. However, individual particles have equal probabilities of moving from one side to the other, no matter the concentration they're coming from. Still, the concentrations will end up being equal if particles are left to flow on their own. If around 6% of the low concentration is moving over to the higher concentration side every second, while around 6% of the higher concentration is moving over to the lower concentration side, the 6% of the high concentration moving to the low will more than compensate for the 6% of the low concentration moving to the high, and in fact add to the lower concentration, because the high concentration contains more particles, so 6% of the high concentration will also contain more particles. Even when equilibrium, when there are equal concentrations on both sides of the membrane, is reached, the particles are still moving constantly from one side to the other, but the amount that leaves one side will equal the amount that leaves the other side, so no concentration change will occur. This can also be put in terms of the energy associated with arranging things, entropy. When the high concentration moves to the low concentration, when the high concentration moves to the lower concentration, entropy increases, while when the low concentration particles move to the high concentration, entropy decreases. Entropy is maximized when the concentrations are equal on both sides of the membrane. The equation for change in entropy that explains this force of chemical diffusion is here, where delta S, change in entropy, is equal to the ideal gas constant times the natural log of the particle concentration outside over the particle concentration inside. However, ions are the particles we're talking about here, and they carry an electrical charge, so it isn't just their concentration that forces them to move one way or another. There's also an electrical force that can cancel out the entropy diffusion force if it's strong enough. This electrical force can be represented as H, enthalpy. The equation for change in enthalpy is the charge on the ion that we're talking about, multiplied by Faraday's constant, multiplied the difference between the voltage inside and the voltage outside. Gibbs free energy equation is an equation that relates entropy and enthalpy and lets you figure out the amount of useful energy that is available for you to do work. If you plug these entropy and enthalpy equations from the earlier slides into Gibbs free energy equation, you get this equation below, where delta G, the change in the energy available to do useful things, is equal to the change in enthalpy minus the temperature in kelvins times the change in entropy. When delta G, or the change in the amount of energy available to do work, is 0, that means equilibrium has been reached. So equilibrium is actually going to happen when enthalpy is equal to entropy, which will not just happen when the ion concentrations are equal on both sides, but will also need to take into account the electrical charge on these ions that are flowing. All right, now how does this relate to the ions in our brain and the electrical signals that neurons are able to send? Well, there's specific ions at specific concentrations inside and outside our neurons at any given point. And these baseline concentrations are what allow the ions to flow in specific ways to try and get delta G to be 0 as soon as the relevant channel proteins open and allow them to. This illustration shows approximately the set of baseline concentrations of different ions inside and outside a neuron in the neuron's resting state. As you can see, according to diffusion forces, the sodium ions, the Na+ ions, would really want to get into the cell, while the potassium ions, the K+ ions, would really want to get out of the cell, if any channel proteins let them. Now, each specific ion concentration will want to flow so that it reaches that ion's equilibrium state. How can we predict how specific ions, like just the sodium ions, will flow into and out of neurons to reach their own specific equilibriums at different interior voltages? What would the voltage inside the cell need to be for a particular ion to be at equilibrium? Let's revisit our Gibbs free energy equation. Remember, at equilibrium, delta G will be 0. Set the voltage outside the cell to be 0 as well, and then restructure the equation to solve for the voltage inside, which we will call the equilibrium potential of the ion, or E sub ion. This gives us the first equation on this slide, where when we solve for the, again, the voltage inside the cell, what the voltage needs to be inside the cell. Because of the constants and because we are using the temperature of a living mammal, and because log base 10 is easier than natural log to work with, we can simplify our equation to the second one on the slide, which will give us our equilibrium potential in millivolts. Our ion concentrations are in millimolar. So if you feel up to it, try solving for yourself. What would the voltage inside the neuron need to be for sodium ions to reach equilibrium? Potassium ions, calcium ions? You can solve it now. For any ion, if you allow it to flow through a membrane, it will want to flow in such a way that the voltage inside the cell will become closer to equilibrium potential. The driving force that drives an ion through the membrane one way or another can be represented by this equation. Okay, in the neuron membrane, so basically like in the neuron membrane at resting state, there are leak potassium channels that constantly let out some of the potassium ions. So the inside of the cell is generally negatively charged at about -70 millivolts at resting state. But this changes when the neuron sends its electrical signal, and let's talk about the shape of this electrical signal a little more, and then we'll talk about the mechanisms that allow the signal to happen. So the electrical signal that travels down the axon of the neuron is called an action potential. It occurs after small bumps in voltage accumulate and reach a threshold, which triggers a massive, rapid positive spike in voltage up to around +40 millivolts, followed by a rapid fall in voltage and a short overshoot period of extra negative charge before the voltage returns to rest at -70 millivolts. So how does this action potential have this specific shape? On the dendrites of a neuron, there are ion channel proteins in the cell membrane that are responsible for receiving neurotransmitters released by other neurons and only open the channels if molecules of the neurotransmitter stick to the protein. These ion channel proteins that open when neurotransmitter binds to them and lets ions flow through are called ionotropic receptors. There are different kinds of ionotropic receptors that bind different kinds of specific neurotransmitters, and each receptor only lets specific ions flow through. Not just any old ion has the shape needed to fit through the channel. There are potassium-specific ones, sodium-specific ones, calcium-specific ones. They can be specific to like multiple ions as well. You get the point. Their receptors are selectively permeable for certain ions. One super important and common neurotransmitter is called glutamate, and it binds to ionotropic receptors on neuron dendrites called AMPA-type glutamate receptors. When glutamate is released from the synaptic terminal of another neuron, the glutamate molecules bind to these glutamate receptors, and the receptors open up and allow positive sodium ions to flow into the cell, bumping up the voltage little by little. This is not the giant spike just yet, but baby steps of upping the voltage. Another type of selectively permeable ion channel present in the membrane of a neuron is something called a voltage-activated ion channel. And the voltage-activated ion channels open or close in response to certain changes in voltage of the cell. What allows the rapid increase of positive charge is the existence of voltage-activated sodium channels. Voltage-activated sodium channels are selectively permeable for sodium ions and open or close in response to specific voltages. There are lots and lots of voltage-activated sodium channels in the neuron membrane, and they have an additional mechanism that makes them even more effective than other voltage-activated ion channels. They have a plug. When the cell is at rest, the voltage-activated sodium channel is closed and its plug is not blocking the channel. When those bumps in voltage from ions flowing through the glutamate receptor proteins accumulate enough and reach around -40 millivolts, a threshold is reached, and lots of the voltage-activated ion channels suddenly open, allowing sodium to flood into the cell, causing a flood of positive charge. This is the rising phase of an action potential, that rapid increase in voltage. And during this time, the voltage-activated sodium channel is both open and unplugged. During the peak of +40 millivolts and the rapid falling phase of the action potential, the channel is still open but is blocked by the plug. Then during the refractory overshoot period, when the voltage overshoots the -70 millivolts of resting state and reaches around -80 millivolts, the channel is both closed and plugged. Okay, now we know what causes the cell to spike to +40 millivolts and not go beyond that. But what causes the falling phase of the action potential and the overshoot? Well, once the voltage-activated sodium channels are closed at the peak of +40 millivolts, voltage-activated potassium channels are triggered to open. And remember, potassium ions want to leave the cell. There are lots and lots of these voltage-activated potassium channels too, and when they open at +40 millivolts, the positive charge of the potassium ions floods out and causes the voltage of the cell to drop rapidly. It actually overshoots -70 and goes down to -80 millivolts before the channels close. Another protein, a sodium-potassium pump, brings the concentrations of sodium and potassium ions to resting state using short-term energy from an energy-carrying molecule called ATP to move the ions against where they want to go if needed. The action potential is complete when the cell is back at its resting voltage of -70 millivolts and its ion concentrations are reset. This process doesn't happen all around the neuron at the same time, but actually propagates along the axon, moving from the dendrites down to the synaptic terminals. Because of the myelin, the signal can actually travel 100 meters per second through the neuron. The myelinated areas do not need voltage-activated ion channels to propagate this signal, which is why the signal can travel so quickly. And the signal only needs to regenerate in tiny gaps between the myelin sheets, called nodes of Ranvier, where the voltage-activated ion channels need to exist. Inside the synaptic terminal, there are little bubbles of fat called vesicles. And inside these vesicles, there is neurotransmitter, for example, glutamate. When the action potential reaches the synaptic terminal, the increase in voltage allows the voltage-activated calcium channels to open in the membrane of the synaptic terminal, and calcium ions flood into the synaptic terminal. The calcium ions actually stick to these vesicles and drive them towards the membrane of the synaptic terminal. When the vesicles reach the membrane, they fuse with the membrane and turn inside out. So the inside of the vesicle faces the exterior of the synapse, and it can release the contents of neurotransmitter into that gap between the cells so that the neurotransmitter can bind to receptors on the dendrites of the next neuron and start the whole process over again. The neuron that sends this neurotransmitter from its synaptic terminal is called the presynaptic neuron, while the neuron that is receiving the neurotransmitter is called the postsynaptic neuron. Now, if neurotransmitter stays in the synapse for too long and builds up in the gap, the ability of the receiving neuron to send its electrical signal is disrupted because the signal will not have a clear stop and a clear start. So the neurotransmitter molecules in the synapse, as the postsynaptic neuron reaches the threshold voltage needed to spike, are either taken back into the sending neuron, into the presynaptic neuron, to be recycled through proteins on the presynaptic neuron called transporters, or they're broken down in the synapse itself through enzymes. This allows the signal to have a clear stop and a clear start. And that's the material for our first class. This was a lot of material, and I'm sure you have questions and areas of confusion, but don't worry, we'll go over this all again tomorrow and work through some examples and problems together. I also want to briefly introduce your first homework assignment, which will be due on September 14th, since we do not have class next Monday on the 7th. I want you each to write a 500-word miniature science fiction story, which has a plot that depends on and demonstrates your understanding of at least 2 of the following topics: ion channel proteins, electrical force and diffusion, and presynaptic and postsynaptic roles in a synapse. All right, see you tomorrow. These were the classes that taught the material that I kind of compiled for this first lecture. Goodbye.